cell division and the cell cycle. Cell division have two types, mitosis and meiosis. Let us talk about mitosis. Mitosis is where you find egg cell, and in meiosis is where you find sperm cell. In mitosis, all other cells that divide. So in mitosis also, parent cells produce two daughter cells, where one divide two and then develop side by side. The important is tissue growth, tissue repair, and tissue replacement. These are important of mitosis. Then let us see about cell cycle. Cell cycle comprises two parts, interphase and mitotic phase. In interphase, they are also composed of three parts, where we find G1 phase, G2, and S phase. So in G1 phase, or you can call it G0 phase maintenance, not all cells divide. So what you have to think about also is in interphase, in G1 also have the new organelle produced, increases protein, and centriole began to replicate. While in S phase, it is where DNA replication occurred, where the DNA replicates into two strands. The DNA polymerase help to the binding of the nucleotides. So here in enzyme helicase help us in the detachment of the strands. So in G2 phase, cell growth continues and centriole replication complete. And what we think about, we are going to talk about in mitotic phase. So let us see, in mitotic phase have the division of nucleus. So cytokinesis also, it is the division of cytoplasm, and here you have to think also about just on the four stages in the mitotic phase, where we are going to start about prophase, and it is where chromosome and chromatids develop. So dear students, as you see here, the prophase, this is where the spindle apparatus are developed and the chromosomes are also tied together. In metaphase, it is where the spindle apparatus develop, where the chromosome lie side by side touching on the spindle apparatus. This is on metaphase. While in anaphase, it is where divided into two parts. So the one part of the chromosome goes on the side one, and another part goes on the side towards the centriole. So dear students, also what you have to think about, it is about telophase. Telophase is where those cells that divide side by side develop and form the organelles on the side, where it also divides in the developing of the cell membrane and cell nuclei. So in the telophase also, the cell starts to develop itself. Dear students, you have to think about also in the mitosis, it is a place where the regeneration of cell occurred, and you have to know that if the daughter cell nuclei starts to divide in the same cytoplasm, it is where you call it the cytokinesis starts to divide. As you see here, phases of the mitosis: prophase, metaphase, anaphase, and telophase. Those are the phases of the mitosis, and you have to know about the cytokinesis. It comes at the last point, where it is the division of cytoplasm, and then the cell occurred, develop side at two part. So dear students, what you have to think, it is prophase. The chromatids are inside of the nucleus, and the chromosomes are not already right on the spindle apparatus. They are scattered in the nucleus and the cytoplasm. So, and the centriole starts to develop aside and nuclear membrane inside of the cell. So, and you have to think about those centrioles. They develop small spindles, but one side on side, and those spindles are the one who involved in the development of the long spindles where the chromosomes lies in the stage of anaphase. As you see, they form what we call equatorial line. Then centromere, it is where the spindle apparatus are one attached to another, and one side is called kinetic core, the point where the spindle apparatus attach on the anaphase. As you see, those one side detached, and the one spindle, one centriole detach the chromosome into two parts, then they form that form of nucleus. So in the next step, which is the telophase, it is where the cytokinesis start, and if the cytokinesis start, it is where there is the division of the cytoplasm, where they develop into new daughter cells. And mitosis develop two daughter cells, whereas in meiosis they develop 4 daughter cells. Dear student, I think you have heard about the mitosis and how it develop. In animals, they form cleavage fallow on the cytokinesis, and in between those 2 daughter cells, then they become mature. While in plants, the cell wall, they make what we call phloem. They don't make the fallow, but also they detached into daughter cell by making cell plate. So just think about also the in prophase, the nucleolus disintegrate and nuclear membrane break down. So the spindle fibers here start to appear. So in prometaphase, the spindle fibers attach to the chromosome, and chromosomes condense. While in metaphase, the chromosomes align at the metaphase plate, and in anaphase, it is where the centromere divide and sister chromatid move to the opposite pole. And what you have to about in the interphase, it is where you find G1 and S1 and G2, where we said that in G1 it is the first growth, and S2 it is the DNA synthesis, and G2 is the second growth. So this is the where there are some exchange of the 46 chromosome is duplicated by the cell, and also the chromosome condensed, as I said, are division of the arterioles. The cell double check the duplicated chromosome for the error and makes n need replay. So there are some adult cells that do not divide. These include brain cells, nerve cells, and eye lenses. The reason why the brain cell and nerve cell and eye lens that do not divide, this is because they can't take result due to its specific function of the body. Most eukaryotic have between 10 and 50 chromosomes in their body cells. Human have 46 chromosomes. Then a chromosome is formed from complex of DNA and a protein called the chromatin. So dear students, thank you for watching me and following me about this module on the cell division, where we see mitosis and its processes in addition with the cytokinesis. So I would like to request you to subscribe, comment, share, and